Hello everyone, and welcome to the very first lecture that we are going to have in Biology 1. I am pausing my voice a little bit because I am trying to find all the information I need to pull together here to make this work, and this should actually now be visible on your screen. All right, so on your screen you should be able to see a picture of. If you don't know what that is, that's an animal cell. You should be able to see that at this point. Let me focus it just a little bit for us. And as I said, this is going to be the very first lecture for Intro Biology 1010 or Intro Biology 1. Most instructors actually start with chemistry in this course, but I don't find that that's a great place to start. Some of you are already gasping now that you found out that chemistry is in this course. It's actually very little chemistry. If you have had any chemistry at all in the past, probably even early high school or late middle school, you have had the chemistry that we're going to be doing, so it's not very detailed. But I prefer to start here because you took biology because you had to have a science, I'm guessing, and you probably were told it's the easiest of the sciences to take because you're not a science major if you're in this course for the most part. My goal in this course and Biology 2 is to give you some basics about science that hopefully will open your mind a little bit more to science, and whether you ever really love it or don't love it, at least you have some better understanding of it. I try to make courses as practical as I can. That is harder for this course in Biology 1 than it is for Biology 2 because Biology 2 is actually human anatomy and physiology. So that one's really easy. We talk about a lot of diseases and real practical things. This course is a smorgasbord of sorts. Because we do cell biology, we do genetics, we do plants, DNA. I think you'll find that part interesting. Some people really like the genetics as well, but it's just a sampling of a little bit of everything. So when colleges designed this particular course, just so you'll know, they designed it basically to bring everybody to the same point to move on into Biology 2. By that I mean some of you listening to my voice have had probably more biology than you'll have in this course. Maybe not quite as much depth, but you've had more. Some of you have had zero biology. Some of you had biology 10 years ago. There's just a lot of different backgrounds. So that's what this course is about. So expect it to be relatively general, but a good foundation to start with. So as I do these video recordings, I try to limit them to an hour or less. I know that makes you want to pull your hair out by hearing that, right? But I try to limit these so that they don't get overwhelming. But obviously, you can start and stop as many times as you like. Take breaks as you like. So what you'll need for these is typically just your lecture notes, because what you're seeing on the screen right now, if you're brand new to my class, is what you would see if you were face-to-face with me in the classroom. You just would see me in addition to the screen. I prefer you see the screen. So that's what I'm doing. This is the same thing I project. I do not use PowerPoint slides, otherwise this would be a voice over PowerPoint presentation. I don't use them simply because I don't think they're the best way to approach an intro biology course. Now we have PowerPoints, of course, that we use in the lab, but not in the lecture. So trying to give you all that information, background information, so you'll know what to expect. So bring your notes, watch the video. After we do the videos, however many videos are in one given module, I will also do a review video for you, and there are a couple of types of reviews that you'll have available to you. So that's kind of how this course is going to work. I'm not going to go into the details of grading, etc., on this recording. If this course is totally online, then I will do that in a separate recording. So we're ready to jump. Let's get into cell biology and look at the general characteristics of a cell. As I mentioned, this is an animal cell that you see on the screen in front of you right now. This, if you've never seen one, most of you I'm sure have seen this as well, is the plant cell. So as we go through, I will actually have to choose one or the other to kind of focus on, I guess, rather than jump back and forth all the time. I will actually center my attention on the animal cell, and then we'll add the things that the plant cell has that the animal does not have. Because here's the bottom line. The plant cell has everything the animal cell has plus 3 other things. And one of those is obvious as you look at this picture, and that's going to be that big central vacuole in the center. So I think it's easier. I mean, yes, I'm kind of biased to animals, because if you don't know already, I'm a veterinarian by profession, but that's not why I'm doing the animal as my main focus here. It's just an easier way to look at it. So we'll do animal plus and call the plant the animal plus story. I'm fascinated with plants and what they can do. So I'll be going back and forth with these pictures. I just wanted to kind of get you acquainted with what to expect, what you're going to be seeing as we go through this. All right, so your notes, you found them in content, should look like this. I have autofocus on, obviously, and we'll see how we kind of roll with that. I can turn it off if I have to. Sometimes it works better with autofocus, sometimes it does not. I'm sitting in the library right now recording these during the summer. I record from home. I record from anywhere that I get internet connection, basically, the back of my car, the side of the road. I've been pretty much everywhere with this, just because there's so many of them. But we're just going to work our way through this and get our basics about cell structure and function. All right, so cell, what is it? Definition there, the basic unit of life, or the smallest unit able to maintain life. That definition is really centered on taking a step back and looking at an entire body and saying, how are we going to break this down at different levels? At the lowest level, you know as well as I know, you are chemical. You are molecules, you are atoms, and that's why there's a little bit of that in this course later on. I put that later in the course. I've already told you one reason why I don't like to start there. The other is because I put that later in the course because I actually use it to talk about nutrition. That is something very few instructors do in this class, but it's the most practical thing I can do with that. But at your basic level, you know that you're made up of atoms. Once we get those atoms together, we make molecules, we make compounds, etc. Then the next level, so to speak, is going to be the cell. So saying that the cell is the basic unit of life is not trying to take it to the atomic level, but it is trying to designate a group of atoms we can put together that are going to have a specific function. A cell in your liver has specific functions. They're not exactly the same functions as a cell in your pancreas or your heart or your kidney. So saying that we're a basic unit of life and able to maintain life assumes that you understand with this definition that the cell requires nutrients to be delivered to the cell, obviously via the blood, and waste products to be taken out from the cell. So maintaining life means maintaining life with support. All right, so why do we have this kind of at levels then? Well, the atomic level, then we go to the cell level, because we're going to take cells, and this becomes a biology 2 topic, actually. And that's got a biodiversity title to it. When I say biology 2, that's what it's been called for 20 years. When you get ready to take that class, or if you've had it already, it's called biodiversity. But we take the cells and we're going to put them together in tissues. And a tissue is just a group of related cells that are close together that perform the same function. Connective tissue, nervous tissue, etc. And then we take the tissues and we organize them into organs. Everybody's familiar with that, your heart, your liver, your lungs. The organs then become part of organ systems. Your cardiovascular system is your heart with all your blood vessels. And that's kind of the levels of organization that we talk about in the human body. Obviously, the plant body is going to have some similarities and some differences, but that's the basic idea. I just wanted you to kind of capture this definition by what it's saying and what it's not saying. As the case may be. That's not a test question. I just want you to know why we're starting with the cell. Because realistically, as we go down through here, the only way you're going to understand an animal body or a plant body is to understand this basic cell. And we're going to take the cell apart, just like you would open the hood of a vehicle to look at the parts. We're going to open the cell to look at the inside to understand it. All diseases are diseases of the cell. All function of the body is a function of the cell. So it's kind of the place everyone has to start. I teach anatomy and physiology as well, and my nursing students are notorious for coming to the first day of class and wanting to talk about heart disease. And I have to slow them way down and say, no, first thing we have to address is the cell, because you live by the cell and you die by the cell. That's your basic unit of life. And you're probably thinking at this point, oh my word, she's been talking for 20 minutes and we have one line. No worries, it's going to go faster. All right, the second line there is on there for the purpose of the final exam. It may or may not show up on my exam, but that's what was put in there. There are many parts. You may be familiar with the cell theory. There are many parts to the cell theory, and I'm not going to go through all of them. But the only part that they chose to put in this course was basically the follow-up for the definition of the cell. Every cell came from another pre-existing cell. That's kind of common sense, right? They don't come from nowhere. So you started as a single cell. Yes, Everybody's in agreement, I hope, out there. You had dad's sperm, mom's egg joined into a single cell, which made you a 46 chromosome individual if you're human. And then from there, one cell became two in a matter of a few minutes. 2 became 4, 4 became 16. You had exponential growth. That's really what this is saying, is that all the original material had to be there to create the cell. If you're thinking, what about plants? If you don't know this already, they also have sperm and egg. So it's the same story. Before you actually left mom's oviduct or fallopian tube to drop down into her uterus, where you would spend the next 9.5 or 10 months or so, before you actually dropped into her uterus, you were already 1,000+ cells in the morula and blastula stages that you go through to get there. So just growth, basically, is all we're saying. You started as one, and you will continue throughout your life to make new cells, obviously. All right, now because this is an intro biology course, which is a very general biology course, we will be talking about both prokaryotic and eukaryotic cells. Now, here's where the main test material starts. Like I said, you may or may not have a question on cell theory, but this is where everything really starts. And if I were you and I was studying this, I would study it just like it is, like in a chart side by side, be able to tell me what is the difference in the prokaryotic cell and the eukaryotic cell. Now, notice the asterisk right here. The asterisk goes with, and I think the autofocus is going to have to go. It's going to drive us crazy. Hang on just a second. Let me turn that off. Every time I breathe or move my hand, I have another little focus opportunity down here, but I have to do it manually. I think that's going to be better, though. The asterisk that you see here goes with the asterisk that you're going to see down here. I try to move slowly on this video because I am using a portable video camera, and I realize that it's kind of jerky and it doesn't always flow well. But what I'm basically saying with this eukaryotic story is that everything else we're going to talk about today, tomorrow, in this whole module, and most of the course will be eukaryotic. We will come back at the very end of the course and visit prokaryotic. But right now we're talking about eukaryotic. We have to get our comparison down, yes. You do have to know that for test purposes, yes, but that's what the asterisk is about, and that's what our focus is going to be. So let's go ahead and define them if you don't know what they are already, and then you'll understand why we're going to be doing primarily eukaryotic. All right, so you see the prefix pro, which you see down here, it means pre, and over here the prefix u, if you don't know already, and it means true. So the karyotic, obviously you can see, means nucleus. So prokaryotic cells, when they first discovered these in the 1800s, they had very unsophisticated microscopes. And with these very unsophisticated microscopes, they literally thought this prokaryotic cell had no nucleus. And in essence, that is true. But the problem with that statement is that you start to assume with no nucleus that there's no DNA. And that part is not true. So make sure you're understanding that. If you have some background and you know that DNA is stored in the nucleus, when I say there's no nucleus, you're going to make a wrong assumption here with a prokaryotic cell. They simply couldn't see it. Their microscopes were not sophisticated enough to do that. So if you come on down here, you see that there is DNA. It's in what's called a nucleoid region, and I'll show you a picture of that. What that amounts to in a very unscientific description is a wad. Basically, the DNA is just tangled in the middle of the cell. So it doesn't have any membrane around it, doesn't have any boundaries. All right, if you come over here to the eukaryotic cell, DNA has a double membrane nucleus and all of its organelles. Organelles we will define in just a minute and discuss, but organelles are the parts of the cell. Just like I said, if you were going to look at that engine to find out how it works, you're going to look at the parts. If we're going to look at a cell, let's look at the parts. So what we're saying over here is the eukaryotic is a much more sophisticated cell, and all of its parts have very protective, very specific membranes around them. Over here, we have very few organelles, but we do have some, but they have no membrane and no real grand organization. So what you see at first glance, your reflection is very simplistic versus very complex. And the only thing I haven't mentioned, well, a couple things I haven't mentioned here. Let's go here. Uni, most of you know that means one, like a unicycle. One cell. These can exist. They don't have to, but they can exist on their own. Multi, obviously meaning many cells. If you look at eukaryotic organisms, they can only live as clusters of cells. You have trillions and trillions of cells. Your cell does, if I pull a cell out of your body and throw it on the tabletop, it dies in minutes. right? It can't live as a single cell organism. So again, it's an idea of complex versus simple, but it's important to understand your complexity comes not only internally with how each cell is made, but how many cells you group together for the whole organism. So that's kind of a same idea on two levels, if you want to say it that way. All right, who are the members? And this may be the thing that helps you the most. I know I've skipped around a little in this, but I wanted to kind of give you the overview of it. Who are the members of each group? Here we have bacteria and archaea. You're welcome to mark out the word archaea if you want to. Archaea means ancients, archaic. You can kind of see it in that word. Ancient bacteria and bacteria, as long as you know that bacteria lie here. And bacteria are the only prokaryotic cells. Very simplistic. Everything else is going to fall over here. The plant cell, the animal cell, et cetera, et cetera, et cetera. So now probably that's a little bit easier for you to start to grasp. Okay, I can really see now the difference in these categories because I know there's a remarkable difference in my human body and a bacterial body. All right, something that I typically ask in class, and I really like to get into this discussion, however, since I'm not looking at you in the classroom, but you're having to listen to me instead, but I want you to ponder anyway, is where does the virus fall? Because virus is not mentioned there. Right. So think about that for a minute. I'll come back and answer the question in a minute, but just think about where would a virus be? And unfortunately, in this day and time, virus is all you hear. Corona just happens to be one of millions of viruses. I'm not going to go into the specifics right now of coronavirus, but we will talk about viruses a little bit later in the course, kind of toward the end of the course, and we can get a little bit more into that if you like. Or if you're like me, you're probably tired of hearing about it. So, but where do viruses fall? Are they prokaryotic or eukaryotic? All right, so think about that for a second. Let's do a couple of pictures here to kind of get this idea in our heads about what did this double membrane thing mean anyway? What was it all about? What's this organizational story? All right, well, you've already seen this picture, so we've already seen the eukaryotic cell, and I could certainly use the plant cell as well. Many organelles. They're going to be floating in what's called a cytoplasm. That's just here in a second. We'll see that in the notes. So everything that you see here, and you can't really in this picture necessarily see a double membrane. This is obviously just an artist drawing. You can kind of capture in a couple places, but I could show you another picture. It doesn't really matter as long as you understand that each organelle has a specific covering, and this is a very organized entity. All right, so let's take a glance at a bacterial cell kind of as a compare and contrast story. All right, and then my camera just a little bit down here. Now, this is a very bizarre bacterial cell, and the reason I say that's because on each end it has, you'll see the word flagellum there, but it has flagella. Sorry, I had to pause. My throat's already dry. It's not typical that you'll find a bacteria with flagella on each side. Otherwise, that's what they use to swim, just like the human sperm. Otherwise, they would just kind of spin in circles. Anyway, ignoring that, beyond that, I'm not interested in the external features of this bacteria right now. I'm more interested in what you see on the inside. Now, think about the picture that we just left. All those organelles, I told you they had this fancy double membrane on them, well covered, well organized. What have you got here? Well, you got what's called the nucleoid region. You saw that on your notes, which I told you was unscientifically a wad. This is your DNA. It just doesn't have a nice wrapping around it. And what else do you see? Not a whole lot. Especially not compared to the previous picture. You're going to see some ribosomes. Cytoplasm is just the liquid. We see that word. You're going to see some ribosomes, and then you're going to see some outside wall coverings. So there it is. Prokaryotic, very unsophisticated, but very capable of survival. Over here, you see a similar story. You just add chloroplast to it. So same kind of idea. We do have bacteria that photosynthesize. You probably have heard or seen in East Tennessee what we call pond scum. It's that nasty green stuff that you see in the ponds or any stagnant water. Those are all photosynthetic bacteria and algae. Yeah, as you can expect, they have the perfect setup for life. They sit in stagnant water, so they have a water source, and they absorb sun, and that's where they get their energy. So there's other places we can find them as well. But there are bacteria that do photosynthesis, and then some that do not. I do not consider bacteria a bad thing. Most people, as soon as you say bacteria, they think, oh, it's going to cause diseases. I would say the majority of our bacteria actually are not pathogenic. You have bacteria that cover your body, that are all over the outside of your body right now. It's a staph organism that are there to protect you. You have bacteria inside of your colon that are there to protect you. There are a lot of good bacteria out there. So when you think about bacteria, don't go automatically. Don't let your default mode be, oh, it's a bad thing. I do want you to, and again, this is a discussion I like to have face-to-face because I love to see reactions to this question. I do want you to appreciate the survival capability of these organisms. And you can capture that really quickly if you just think, if we had a major environmental event, maybe you think nuclear war, whatever comes to mind for you, who's left standing, you or a bacteria? That's a pretty easy question, right? What is necessary for you to survive? And you may think, well, that's basic oxygen and sugar. Yes, it is, but think about true survival of your human body or the animal body versus survival and reproductive capability, we'll throw that in, of these organisms. I can put one of these in a little petri dish on Friday, come back on Monday, and there's 1,000 of them. Can't do that with a human. You're probably going, and I'm really glad. But if you think about their capability, their simplicity makes them more capable of survival. They just don't have a whole lot of needs. So you got to respect these bacteria for that. Now, the question that I like to ask in class that I mentioned but didn't actually come forward with the question was concerning things that you may have heard of called superbugs. And I think everyone is familiar with antibiotic-resistant bacteria. So the question I would ask you if you were in front of me is, if I walked into your house today, and maybe I knocked, maybe I didn't, maybe I just came on in, walked into your house today and said, excuse me, can I use your restroom? And I went into your bathroom, and not that everybody keeps their stuff in their bathroom or has necessarily a medicine cabinet, but let's go with that example for now. And I opened your medicine cabinet. Would I find, this is my question, would I find any unused antibiotics that were out of date or that should have been taken? Like, I'm not talking about ones that you're taking at the moment, should have been taken last week, last month, last year. Most people are probably smiling, going, yeah, you probably would. Have you ever stopped taking antibiotics? Sure. I think, well, a few people haven't. A few people are good at this, but you know, you take an antibiotics because you had some type of infection, and day 3 you feel better. By day 4 you forget. Right? And so that little stack of antibiotics just kind of sits there, and you push it aside, and I'll get better. And your body usually comes in and finishes out, you know, destroying the pathogen, et cetera. But understand that that's one of our leading problems for antibiotic resistance. And what does antibiotic resistance mean to us? It means that we give these bacteria a sample of our arsenal, how we can eliminate them. And if we give them a sample but we don't kill all of them, then you've heard the word, everybody knows they mutate, they change. Viruses do the same thing. We're not treating them with antibiotics, but they do the same thing. So when you give them continuously, think about how many people, millions of people in the world do this, continuously we're giving them samples of what we're going to use against them, they eventually can, in some cases, become our superbugs. If you're not familiar with the superbug term, it just means something that is beyond our current capability to combat when it comes to the bacterial world. We don't have an antibiotic that will treat it anymore. It is now resistant to all of our good stuff. So really, medicine in this field is a race between how fast can we develop new antibiotics, what can we possibly do, as compared to how fast can these bacteria replicate and adapt to their environment and mutate and change to where the antibiotics are no longer capable of killing them. So just be very aware of that. Now, I have a lot of people who say to me, no, it's not that. We've got antibiotics in our meat sources, in our milk sources, and this, et cetera. Other people, and by the way, before I just kind of scoot on over that, because I just spent a lot of time in class talking about this, I was a dairy veterinarian for about 15 years. And I can promise you that in this country, your milk products are safe. I have watched dairy farmers cry when the milk goes down the drain because it's thousands of dollars when they have to dump a tank of milk. They are extremely cautious about treating their dairy cows with antibiotics. And we actually, that was part of my job, If I gave an antibiotic to a cow and she was a milking cow, if the withhold time on her milk was, say, 7 days, we went 14 to 21 days before we ever let her milk go into the tank for human consumption. And then it was checked 3-4 times before it left the farm. It's checked again when it gets to the factory, etc. So I feel confidence. I feel like milk and cheese and those dairy products are very, very safe. When it comes to meats, I have no point of reference with the poultry industry. I mean, I know there's inspection and all that in place, but I can't personally speak for that. With the beef industry, the only problem that I have seen arise, and I don't think this leads at all to antibiotic resistance in people, but it was not something that the veterinarians could control. I would never, ever send an animal to slaughter if they had antibiotics in them. I did the best I could with that, but farmers can go down to the co-op or Tractor Supply or whatever, and they can buy a food with a low-level tetracycline in it. Now, I highly discourage this for many reasons because we didn't want the antibiotic resistance in our animals either. And some will do it and some will not. The level is extremely low. I can't speak with the confidence I do for the dairy industry, but I can speak with significant confidence that your beef products are safe. Didn't do a whole lot with swine. I did work for some of those farms, and they were very safe at the time. So really, as far as personal experience goes, I would say most of that is a safe bet. Now, some of you may disagree, and that's fine. I mean, if you want to go research that and look that up and find, aha, we found this and this, that's fine. I still don't think it is the big player in the story of why are we as a human race going to have superbugs? Why are we facing this resistance problem? It may be a player, and you may find evidence of that if you're interested in researching it, but it's not the big player. The big player is individual people not finishing their antibiotics and you know this one, number 2, you know this well, over-prescribing antibiotics. And I'm not being critical of the medical profession, but it does happen. It happens in my profession as well. So the mom comes in with the kid, and the kid's got a virus, and the mom insists that the doctor prescribe antibiotics. And you may think, well, the doctor should just say no. This is Mama Bear, okay? Let me just clue you in on this. Her eyes are spitting fire. She means business. Something's going to have to happen here. So I don't know. I don't have an answer for that. It's frustrating on all sides of the table, but I'm just telling you that you know as well as I know, whether it's that scenario or some other scenario, antibiotics are overprescribed. So not finishing them, overprescribing them, I think are 1 and 2. The animal source may or may not play into your life. I just want you to think about what you can do about that. And what you can do is maybe not ask for those antibiotics quite as often if you don't have to have them. And if you do use them all. Now, I don't want to leave that statement dangling in your mind without saying there are some infections. There's no games. Strep throat is one of those. We play no games, no hesitation, antibiotics, hard, fast, best you can get, because strep can go to your heart and it can cause damage to your heart valves. So don't take that as a total blanket statement. You have to understand what I'm saying. There are some bacterial infections that you can fight off on your own. That's what your body's immune system is made for. There are others that we don't play with. All right, so always seek the advice of your medical professional, obviously, but just do your part. Take those antibiotics when you do get them. All right, how'd I get on? Was that a tangent? I'm sorry. Remember, I said I try to make this practical. I want you to get something out of this class that you're going to walk out the door and say, oh, well, yeah, at least I've thought about this. And some things may intrigue you. You may go research on your own to see where we're going with that, because it is your future. Understand that your generation is going to encounter this more than mine did. And mine encountered it more than the previous one because we have more and more medicine, more and more intervention. So we're going to continue to have this. So remember here, our big point, though, as far as this class goes, was simplicity, but very sustainable, very survivable. These are amazing organisms. All right, so that's your prokaryotic and eukaryotic differences. Now, the question that I left you with a moment ago was about viruses. Where do viruses fall? And I would love to jump in here into a description of viruses, but I won't do it for the sake of time. Viruses, the answer to the question, did you get it right, is neither, or neither, if you say it that way. A virus is neither prokaryotic nor eukaryotic. Why? Because a virus is not a true cell. A virus cannot reproduce on its own. And you may be thinking, well, it can sure wreak havoc on the stock market. Well, that's a whole political story. But a virus has to get into a host, whether that's you or the dog or the cat or the plant or whatever. It has to get into a host cell. It uses the host cell DNA and all of its cellular machinery to remake itself. So I'm not saying that it can't be contagious and transmitted and all that. What I'm saying is it is not a true living organism. In order to be a true living organism, you have to be able to have both DNA and RNA. You may or may not know what RNA is, but basically what that statement says is you have to have DNA and the ability to use it to reproduce yourself. RNA is how a cell makes protein and sustains itself and reproduces its parts. So that's why you do not see viruses listed here, because they're not true cells at all. And this topic is about cells. All right, so we'll come back to viruses. Maybe that intrigues you a little bit. Do understand that when you share viruses with people, you share parts of their cell. That's pretty scary. And we'll talk about it more later. Okay, so moving on down, make sure there'll be test questions on the difference in prokaryotic and eukaryotic, so make sure you know the differences. Let's continue with all eukaryotic cells. We'll come back to bacteria a little bit later. I would love to spend a whole semester in microbiology and just do bacteria, but that's not what this course is designed for. So let's go down into what I've got listed as cell biology. The rest of this printout, for the most part, there's a little bit of a summary at the end, deals with these organelles, these parts. I already told you that's how we're going to look at it, because it's the best way to understand it, by understanding its parts. So what does a cell have? You've already seen this. Let me flash the picture back up here just so we can talk about those three words: plasma membrane, cytoplasm, and organelles. All right, so plasma membrane, the outer covering. Some of you may call that a cell membrane, which it's okay. It's not technically true, because a cell membrane, we would actually come over here and start talking about the fact that a plant cell has both the plasma membrane and the cell membrane outside of that. So technically, the cell membranes are in the plant on the outside. But if you use that term, you're not getting zapped here. It's not that kind of deal. So your outer boundary is your plasma membrane. We will spend quite a bit of time talking about, well, actually the whole next handout talking about things being transported back and forth across that membrane, but it's your covering. Cytoplasm, mentioned to you before, it's on your printout, there's the word right there as well. Cytoplasm is a jelly-like fluid. Matrix is a term that we use, and I use the word jelly-like because I don't want you to think it's like water. It's much thicker than water. It's more of a molasses consistency. But what does it do? It holds, it suspends, it floats, it moves all the parts, which are the organelles. So what does the cell consist of? A covering, a semi-fluid matrix, a thick fluid matrix, and then the individual parts of the cell. That's what the cell actually consists of. Same thing with the plant cell. It just adds some outer boundaries. And like I said, a couple extra organelles. 3 extra organelles to be technical and correct. All right, so that's your cell. We are going to start the next part of your handout, beginning with organelles, starts with talking about the nucleus. All right, so I think everybody is already seeing it here. Here's the nucleus, and you already know, we've talked about it in this video, you probably already know that this is where you're going to see your DNA. So nucleus is always described as the center of the cell, the boss of the cell, the control area of the cell, if you want to say it that way. Now, if you'll notice on your printout, something that I want to point out as we talk about DNA. And when I say DNA, a lot of different things come to mind. Again, if we were face to face, we would talk about, I say DNA, you say what. Some people say double helix, some people say genes, some people say forensic crime scene evidence, whatever comes to mind for you. We all understand that DNA is your genetic makeup. So it's your chromosomes, if you want to say it that way. That's another term you can use for it. It stands for deoxyribonucleic acid. We'll get into that in more detail a little bit later in the course. But your genetic information can take on one of 2 forms. Now, this chromosome word, I would dare say everybody listening to this video knows what a chromosome is. Chromatin may not be quite so obvious for you. What is the difference in chromatin and chromosomes? Now, understand when I'm talking about this, that the DNA itself is not changing. It's just how it is presented that is changing. And you may want to make a couple of notes for yourself on this particular part of what I mean by chromatin being uncondensed or decondensed and chromosomes being a condensed form of DNA. So let's look at a picture, and then I'll come back and write on the printout what you would need to write down. So I try to write on these printouts anything I would put on the board if we were in the classroom. So don't worry if I say a word that you've missed something. If it's important, I'll make sure it's in print in front of you. All right, so let's go over to this picture. All right, for those of you who think double helix, that's what comes to mind when I hear DNA. There's the double helix for those of you who think about that. All right, this is chromatin, T-I-N. And this is what I'm going to call thread-like, decondensed, if you want to use that term, form of DNA. This is the form that the DNA is in inside the cell 99% of the time. And when your cell needs to make protein and do its everyday activities, this is how we see DNA. However, you are also familiar with the term chromosome. So when a cell is preparing to divide, I've got a skin cell that's going to divide into another 2 skin cells. In our everyday growth and repair of cells, obviously we are making trillions, just red blood cells and white blood cells alone, trillions are coming out of your bone marrow every single day. As we're making these new cells, we have to take this DNA and put it into a form that we can move it and manipulate it. Because if you think about this for a second, I've already said the human chromosome number is 46. If I'm going to take a cell with 46 chromosomes and create another cell, the end result is 2 cells with 46 chromosomes. So in the process of doing that, I have to copy all the DNA and then I have to sort it. Well, if it was all stretched out like this, it would simply tangle. I mean, I know that sounds just a little too practical, but it's real. So condensing DNA into a chromosome, the purpose of it is so that the cell can divide and not break its DNA in pieces and get tangled up. And you're probably thinking, oh, that's way oversimplified. Not really, actually not really. That's what goes on. So there are changes in magnification as we go down through here. That's not really a big deal for us, but what's going to happen is a cell is not dividing. The DNA looks like this. When we get ready to divide, you're going to take this, change your magnification, and make this little string down here the same as this in your mind. Just change your magnification. So we're going to take this and we're going to roll it around these things called histones, not a word that you need to know. Histones, etc. You just continue to wrap this up like balls of yarn until finally you get down here to the chromosome. It's still DNA. It's in a different form because it's been wadded up, rolled up, stuffed together so that I can move it around. Because when you get ready to make a new cell, understand that half of this chromosome is a complete double helix of DNA. The other half of this chromosome is another double helix of DNA, and we put them together. So when I get ready to take one cell and make 2, this side of this chromosome will go to one cell. This side of the chromosome will go to the other cell. That's how they get exactly identical genetic information. The skin cell that you're making right now needs to be identical to the skin cell that it came from. Otherwise, you grow a liver on the side of your arm, which actually financially would work out for you because somebody would chop it off and use it, right? All right, so, but we understand cells have to act just like the cell that they came from. So that's what we see here in terms of our replication. And that's the purpose for having the thread-like form versus the condensed form. That's the purpose in talking about chromatin versus chromosomes. So seeing that we haven't changed anything about your genetic code, we've just managed it. We've just put it in a form that we can work with it. All right, so how would this be described on test? DNA can come in one of 2 forms. Chromatin I would describe as a thread. My pencil's going to write. A thread-like form of DNA, okay, is going to be your chromatin. And this is when a cell is not dividing. I don't know how I ended up with a pencil instead of a pen, but that's what I have. Okay, that's the chromatin story. A chromosome is going to be what we would call a condensed form of DNA. And I'm going to put the word form over here just to make those match. All right. So thread-like form versus condensed form. And this is when the cell is dividing. Dividing, as some of you know, is called either mitosis or meiosis, whichever. And we will go through those processes. It is part of this course. We will go through those. Okay, but there's your difference. Now, the reason I'm having you learn that now is because it makes it a whole lot easier for you later. This first test is actually very simple, comparatively speaking, because all you're doing is learning parts and what they do, if you don't already know them. So anything I can add in here to make the future parts of the course a little bit easier, then that's what I do. All right, so there's your control center of the cell, your DNA. Another thing that's in the nucleus that's not DNA, but still inside the nucleus, is this thing called a nucleoli. Or you may see, instead of that I, you may see a US ending. US is singular, I is plural. So they have these structures that make ribosomal subunits. And you're probably going, well, that was really helpful. Okay. Anytime you see the word ribosome or ribosomal, go ahead and get this tucked in your head, that means protein. That means protein is going to be manufactured in a ribosome. So what's happening here, notice that there's a plural, subunits. You're making what's called a large subunit and a small subunit. When they come together, they will be a ribosome. You're going to get this in just a few minutes in this next section. I'm just trying to kind of prepare you for this is where it all starts. So what we're saying is the nucleus makes these two pieces of a factory. There's a small piece and a large piece. When they come together, they are the total factory. The factory is called a ribosome. That is where protein is made. And realistically, the most important reaction that a cell can do is aerobic cellular respiration, which means making energy. But how a cell is identified is its protein story. Protein, you'll hear me say this over and over and over and over. Protein equals function. If I walked up to you and asked you what your job is, that's your protein. And you may be going, I hate my job right now. Well, regardless, that is your protein. For example, what cell in the body makes insulin? There's only one cell type that makes insulin. Some of you answered, some of you are going, I don't know. Pancreas. All right, so the function of the pancreatic cell, there are many functions, but the one I'm focusing on right now, the function of the pancreatic cell is to make a protein called insulin. Protein equals function. All right, so it's a very, very important concept, and you'll find that most of the rest of this lecture today actually deals with protein because it is identity, is what it boils down to. All right. Okay, so again, that's kind of leading you into some other stuff. Let's look at the picture. Let me jump back to that because you may or may not have noticed it when I had it up here before. Okay, so our nucleus is the purple blob, right? Doesn't include this part, obviously. I hope that's obvious. Here is the nucleolus, or if you have several of them, nucleoli. So you can see kind of the stringy DNA in the background. So DNA is in the nucleus, the nucleoli is in the nucleus. And then the last thing mentioned there is going to be its double membrane on the outside. All right, so what is unique about this double membrane? Well, you can see that this is a cutaway picture, of course, but it's got holes all in it. So they're very specific pores that you see. You can also see that the pores, in some cases, are continuous with these series of passageways over here called the rough endoplasmic reticulum, which is the next thing we're going to be talking about. And some of these are not continuous with that, and they just empty out into the cytoplasm. So this nuclear membrane is a double membrane because it's a eukaryotic cell. All I really want you to know about this, let's scoot this up just a little bit, we're right down here. The membrane or the envelope, whichever word you want to use, around the nucleus does have pores. And what is on this printout is what I want you to know. And I'm saying that twice because if you were to go Google or research the nuclear membrane, you would find all kinds of different discussions about possibilities and theories and these things we know and these things we assume, etc. We don't need to do any of that. I gave you what everybody agrees on. Understand that these pores are regulatory, and that's very common sense because if you know the nucleus to be your control center, what's the last thing you want the enemy to attack? Your control center. right? You're wanting to protect that DNA, then you know there's got to be some pretty heavy-duty regulation going on here. So what is allowed to pass for sure that everybody agrees on is all that I'm going to emphasize here. So protein can go into the nucleus. Ribosomal subunits that we just talked about, they're made up here, they can exit. And RNA, which is going to help with your protein synthesis. We actually do a whole module on RNA. So ribonucleic acid as opposed to deoxyribonucleic acid. It is the machinery of the cell to make protein. In other words, RNA uses the DNA information, the genetic code, to make the right protein because we just said protein equals function. All right, so protein goes in. All you need to remember, ribosomal subunits and RNA come out. So because the nucleus can't make its own protein, it has to import some. But most importantly, we're still seeing the nucleus as a control center. And it is sending out pieces of a factory and what works with the factory, the RNA, to make that protein. This is a protein story because your genetic code, your genes, your identities, they're a protein story. Protein is identity. And how does the body work so well? Because the liver reads certain pieces of DNA. The kidney reads different pieces of DNA. They make their specific products. They can all have the same factory. They can all have the same instruction manual, but they're going to make different products. That is the essence of the mystery of life, really, how they can all have the same DNA. I love that. And you may be saying, wait, stop. Well, come back up. All right, did you not come from one cell? Yes? We already talked about it. You know you did. So you had the union of mom's sperm and mom's sperm. I did it again. That is so hilarious. Mom's egg and dad's sperm. I do that in class all the time, and I don't know why. You're probably thinking, well, she is wacky. So we have dad's sperm and mom's egg, and they come together, and you were a single cell. Yes, you were. And then you became two, and we talked about before you even get to mom's uterus, your thousands of cells. Well, understand that in that process, somewhere along the way, every cell gets the same DNA. If you're copying every cell, it's going to get the same DNA. And then you develop into germ layers, and then you develop into organs, and then you become that baby, and then you're born. But you have in every cell the same DNA. If that's the case, why do you have different organs? If that's the case, why is the pancreas the only cell that can make insulin? Doesn't the kidney have the genes for insulin? Absolutely. And your lungs and your brain and every other part of your body. This is one of the most fascinating parts of all science. Who was the first cell that stood up and said, I am the precursor to the liver. Everybody that comes from me will look and act like a liver cell, and they're going to read the instruction booklet the way I do. We're going to read page 5, 10, and 16. And the pancreas next door says, oh, I don't care what you do because I'm going to read page 14 and 37. And I'm going to make my proteins according to those instructions. Do you see what I'm saying? It unfolds as a mystery of sorts. I shouldn't even say of sorts. It unfolds as a mystery that I'm assuming mankind will never understand. We can like mimic the process, we can clone, we can play with DNA, but we didn't create it, and we don't totally understand it. It's very, chemically we understand it, it's very simple. We don't totally understand how one cell can start to divide into multiple cells, and programmed in that first original cell was the instruction and the ability to understand that different organs are going to do different things because protein equals function. I love it. Sorry, I got into that, but I just hope you ponder it at some point in your life and think about there's nothing random chancy about this really, that there's a very specific genetic code, not just with instructions for what one cell is going to do, but with instructions how somebody had to divide off of another cell and become their own entity with the same instructions. All right, I'll leave that alone. I just love it. Okay, so that's your nucleus. Things you need to know, the things we read on the paper there about the DNA, that you have a nucleolus or nucleoli that makes your ribosomal subunits and the characteristics of the membrane. All right, moving on to organelles you may not be as familiar with. Let's talk about these two together because as we look at these two, then we're going to start to see some similarities, but then immediately some differences. The similarities are in the names, RER and SER, the rough and smooth endoplasmic reticulum. Endoplasmic reticulum, I believe, don't quote me on this because I don't remember. I think those came from a Latin word that meant, I don't know the Latin word, but it means continuous passageways or hallways. One is called rough, and this is already on your notes for you because it has ribosomes. It makes it look like it has little studs on it or nails or something sticking out of it. Smooth is called smooth because it has no ribosomes. The surface is smooth. Wow, that was rocket science. But they both are continuous passageways. So let's go back to this picture where we can see that. All right, so there's your rough endoplasmic reticulum. See all the little knots on it? That is a ribosome. Those are ribosomes, I should say. All those are ribosomes. And there's your smooth endoplasmic reticulum. All right, so there's your kind of difference. So they are named for what they physically appeared to be. Now, this was not possible until we got the electron microscope to see what was really going on. Basically, before that, with just the plain light microscope, like we use in our labs here on Walters State campus, all you can kind of see is a nucleus. So once we started looking at these and discovering those, they started naming parts right away, and that was like, okay, let's call it this. But continuous passageways. We've got cutaways on both of these, but they are continuous passageways. All right, so what is unique about the rough endoplasmic reticulum? I've already captured a lot of this one, and it's very easy because anywhere you see a ribosome means what? What did it mean up here? Protein, 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 protein. This is where you make your protein. Now, where it says site of ribosome assembly and protein synthesis, all this assembly word makes reference to is the fact that I told you up here we had two subunits. I had a large subunit and a small subunit, and they were going to come together. Right? Okay, so when they came together, they were going to become the full factory. So all this is saying really is that we're going to make protein as we bring our parts of the factory together. That bringing together of the parts of the factory is a later topic. So if you will equate this in your mind to, yeah, protein is the name of the game here, then you'll be fine. All right, so that's it. I mean, I've already talked about it. I'm not trying to downplay it at all, but there's not a lot else to say about it. It's your identity, it's your protein for that particular cell. All right, so smooth endoplasmic reticulum. We've already said why it's called smooth because it doesn't have the little dots all over it. Notice what's going to happen here. Realistically, if you'll just kind of break this down, I'm trying to make this as simple as I can, break this down into two categories. The cell is going to make its protein here, and it's going to make its everything else down here. How's that for easy? All right, now, some of you know that an enzyme is typically a protein, so don't let that confuse you. There are a few enzymes manufactured here. There are some non-protein enzymes manufactured here. Just remember protein, everything else. So lipids, steroids, there's a lot of other things in this category I didn't want to like belabor you with, but if you need something and it's non-protein, it's going to be made by the smooth endoplasmic reticulum. That is not the smooth endoplasmic reticulum's claim to fame, so to speak. Its claim to fame is the next part, the detox. All right, so when I say detox, what comes to mind for you? What do you think about that? A lot of people say liver, right? Because isn't that your main detox organ of the body? Yes. You may use the word filter instead, which is fine. But everything that comes into your body, for example, everything that you eat, it actually goes from the intestines to the liver first, so we can get some of the toxins out of it, the food additives or whatever that you don't need, the breakdown products that you don't need, so we can get that out before we even send that pool of nutrients through the blood to the rest of the body. So your liver is your primary detox organ. I'm not saying that the liver is the only cell that has SER. All of them do. The liver cell would have more because that's more of its function. So it does detox the cell, takes away the things that we don't want in there. And I'll give you a picture again while we're talking about it. So this again was a smooth endoplasmic reticulum. Okay, as we think about this, again, one of those practical applications I want you to think about. What happens when we overwhelm the cell's ability to detox? All right, and I will go ahead and address the all-familiar alcohol because I'm sure that everybody knows that alcohol can cause cirrhosis of the liver, and it is deadly. Now, before you go, I know so-and-so who drinks every day of his life and he's perfectly fine. He is not perfectly fine. I can promise you he is not. The liver is extremely regenerative and extremely forgiving as long as you feed it. It can be very responsive. You may be familiar with the idea that we can take a liver that is too small for you, not necessarily a little baby liver, but we can take one that's a little bit too small for you. We can put it into your body and it will get larger to accommodate the needs. Very adaptive organ if you give it nutrients. But the true alcoholics, what do they have for breakfast? You're saying it either out loud or in your mind. Alcohol and lunch and dinner. Do they eat much? No. Do they eat well? No. Are they killing themselves? Yes. Now, I am not saying, don't hear this, please. Don't think this is what I said. I am not saying that it's okay to drink as long as you eat well. That is not what I said. There is always damage to the liver with alcohol and the brain, but just focusing on the liver right now. There is always damage there. The only thing that the food does is it prolongs the period of time that your liver can hang on. That's all that it does. It buys you some time, so to speak. And the word cirrhosis actually means scar. And I'm pointing that out because when we actually talk about different forms of liver damage, this one is one of the most devastating because a scar does not regenerate new workable tissue. Look at the scar on your arm or your leg or whatever. I mean, the line may have changed. You may have had a big white line that faded over time or whatever, but the inside of that scar, you know as well as I do, is not living reproductive tissue. It may change shapes because the cells around it are changing, but that scar doesn't grow. All right, and it can't perform any functions. It's a dead spot in your skin. Well, these are dead spots in your liver, and they do not regenerate. The liver can regenerate around them, but eventually, over time, the liver fails. Had a brother-in-law who died from cirrhosis of the liver due to his excessive alcohol consumption. He turned yellow. You're familiar with jaundice, maybe. His skin turned yellow. His eyeballs turned yellow. He got the big ascites, which is the distended abdomen. He was a lawyer, and he kept trying to make a case for, he even tried, I think, to sue the hospital to be on the liver transplant list. Now, he lived in North Carolina. I do not know the state laws on this, so not making this as a quotable or anything like that. You would have to go look this up, and this was many years ago, so I'm sure things have changed. At that time, they told him no. You chose to destroy your liver, therefore we are not going to take a donated liver, an organ donation, and give it to you. We will give it to someone else. And that may be the law here now in Tennessee. I have no idea. Again, if you're interested, go look. But it was I'm just saying it to say that it was a very long, slow, agonizing, painful, miserable death. It is a horrible way to die. And it takes a while. So just so you know. So we talk detox here and we talk alcohol. We are talking something unique, which is cirrhosis. I'm not saying that there's no cirrhosis at all from other things, but the alcohol is the one that does it progressively and consistently. I guess I should use that word. One other thing about the liver, I mean, everything you put in your body, you have a price to pay, right? There's detox to everything except for maybe water. Okay, depending on what's in your water, I guess. But the other thing that I want to mention to you is the over-the-counter painkiller. I want to make sure that you're aware of the dangers of this one to the liver. And if you don't already know which one that is, it is Tylenol, acetaminophen. I am not against the use of Tylenol at all. Just want you to be aware that it is very toxic, deadly toxic to the liver in really high doses. All right, now Tylenol is very different in the way that it works than things like ibuprofen. If you're not familiar with ibuprofen, Advil, etc., those are actually non-steroidal anti-inflammatories, so they are not pain relievers directly. That may be a side effect of theirs, but they decrease inflammation. And when you decrease inflammation, you decrease pain in a lot of cases. Tylenol is a very specific, potent pain reliever. It's also not quite the same as aspirin, so none of those work exactly the same, but the problem is the toxicity. And if you will look on the containers now, this may require magnifying glass because you know how the print goes on those. The Food and Drug Administration in the United States requires every single product that contains Tylenol to have a warning label that it can cause damage to your liver. And it took a long time to get that to happen. I'm old enough to remember those arguments about it, but it's very real, so be very cautious. The problem does not typically come in overconsumption of Tylenol by itself. It comes in consumption of Tylenol and things that you don't realize have Tylenol in them. Like if you're a big cold medicine taker, I challenge you to go out there and find a cold medicine that doesn't have Tylenol in it. Anything that says reduces fever, pain, it's got Tylenol in it probably. There may be one, I don't know. I don't search those shelves in the grocery store, but that's where we actually get people that end up with way too much. They take Dayquil, they take NyQuil, they take Tylenol, etc. The other place is your prescription narcotics, codeine, OxyContin, all of those. I'm not going to go through the whole list, but almost all of those contain Tylenol. And many of those contain 500 milligrams plus of Tylenol. And they do that obviously to reduce the amount of narcotic needed, but sometimes I wonder if that's really wise. I don't know. It depends on your situation, I guess, but just be aware that it's coming in places that you don't think about it being there. So what is the toxic dose for Tylenol? And I'm not actually going to give you that number. I want you to go look it up, talk about it in the next video. I want you to find out how much Tylenol can you take and it's going to be safe for you in one day. And I want that number to be in milligrams. And when I do this in class, I just have us do it kind of as a table and everybody guesses. But it's a number that when we do it that way, most people remember it. It's a number that I want you to have. It's not a test question for those of you just worried about the test questions. It's a number that I want you to have in your mind because you are your family's advocate. I had a very bad situation with my son where doctors were actually overdosing him with Tylenol. And I really had to put a stop to that. I won't tell you that story. But I had to be the one that said, no, this is a toxic level. And it was sad that I had to be the one. Always, always look out for yourself. And always, always, if you have a question, ask your pharmacist. I can't say enough good things about pharmacists. I mean, I know there's probably some wacky ones out there. That's true of any profession. But they have way more knowledge of drug interactions and effects on the body than the average MD, just because it's how they spend their time. I'm not saying they're any more intelligent, but that's their main thing. So always use your pharmacist when in question. All right, so be protective of your body. Don't do the alcohol thing. And then I'm sitting here thinking, okay, so some people just laugh this off. So I'm going to go get drunk this weekend, and then I'm going to go take Tylenol after I get drunk to get rid of my headache the next day. You're pushing all the buttons at that point. At least now if you push the buttons, you'll know that you're pushing the buttons. So no, I do not recommend that at all. But that's what we mean by detox. So taking those toxic products out. All right, the last thing you see here under smooth endoplasmic reticulum mentions the creation of a transport vesicle to deliver the protein that was made up there in the RER. Remember, that was RER, not SER, to our next organelle, which is going to be the Golgi apparatus. So a transport vesicle, and you do need to know the word transport here. A transport vesicle is, for lack of a better word, simply a bubble that we're going to stick the protein in to isolate it. So we're going to take a protein from here over to what's called the Golgi. Now, this transport vesicle can come from here, the RER. It can come from the SER. It doesn't matter. The point is that the protein is wrapped in a bubble of protection so that we can move it to a place where it can be modified. And we're going to pick up on the next video, just because this one's running a little bit longer than I thought, so more intro than I thought I would need to do. You're probably saying more than I should have. We're going to pick up there with that idea of what is the Golgi apparatus and what does it do with that protein. All right, so that is the end of organelles 1, 2, and 3. I think like there are 8 total, so we should be able to, it'll go faster from here. We should be able to wrap these up in one more recording. I hope you have a great day, and I will see you next time.